Hi, and welcome to episode 77 of the Breaking Bio podcast. I'm Morgan Jackson, a PhD student at the University of Guelph in Ontario, Canada.
I'm Stephen Hamlin, postdoc at the University of Southern California in Los Angeles.
I'm Tom Housley. I work at the University of Cambridge in England.
And today we're joined by Dr. Adrian Smith, who is a postdoctoral research assistant currently at the University of Illinois at Urbana-Champaign. Hi, Adrian. Thanks for joining us this week. You want to tell everybody listening in at home a little about who you are and what you do?
Sure. Hi. Thanks for having me. I'm a postdoc right now at the University of Illinois. I study ants. I specialize in ant behavior and ant chemical communication. And I also do a podcast, a science podcast, just like sort of like this one, called Age of Discovery Podcast. And that's basically it.
Let's be clear, there's nothing like this podcast.
That's true. No, that's very true.
It's probably a good thing for the internet.
Actually, I checked out your podcast, and it looks all slick and professional, which, it's another reason it's nothing like this podcast.
Right. Well, I don't know about that. I mean, I have 9 episodes. This is 77, so there you go.
Yes, we win on volume. All right.
Quantity versus quality comes to the forefront again. So you mentioned that you're an ant behaviorist and chemical ecologist, and you just had a paper come out this week, which I think we'll get to in a sec, but can you explain a little bit about what chemical ecology is for those who may not be familiar with it?
Yeah, well, I sort of am a chemical ecologist by default. I really am interested in ant behavior, and a lot of behavior of ants and insects in general is guided by the chemicals that they use to communicate and interact with. It's basically just looking at basic ecology or behavior questions and considering communication through chemicals, which is sort of the default route with which most insects communicate.
So how do insects actually emit these chemicals? What kind of chemicals are they, and what sort of behaviors do they control? Or
yeah, so the type of chemistry that I study and that I specialize in has to do with cuticular hydrocarbon profiles, which is sort of a long term, just to say the waxes that coat the cuticle of an insect. So basically all insects have these compounds. It's basically a layer of wax that's over their entire body that's there, first of all, to sort of waterproof the insect's cuticle to prevent desiccation. So they're constantly there and constantly being sloughed off and regenerated all the time. And for social insects, these chemicals, they do 2 really important things that I study sort of in tandem. They provide a means through which ants can go and identify nestmates from non-nestmates, and they also provide a means through which individuals advertise their own fertility status. So if an individual is reproductively active, her overall hydrocarbon profile changes to reflect her reproductive ability. And so all this communication is happening through a layer of basically 20 to maybe 50 or so individual waxes that change sort of quantities and amount on the insect cuticle, and those changes are reflective of nestmate status and also fertility status.
so I always find it kind of difficult when I've been reading the particular hydrocarbon literature. You're looking at profiles or particular combinations. So how do you kind of pick apart what you're looking for or what you're testing?
Yeah, it's really hard for things that are sort of a gestalt or overall sort of cumulative thing, like a nestmate profile. Nestmate profiles are usually gestalt profiles, so the whole slew of 20 to 50 hydrocarbons really can sometimes all contribute to the overall perception of whether an ant belongs in the nest or doesn't. But then within that, especially for fertility signals, some of these ants and some of these species, when you compare sort of the whole variation of a worker to what's in a queen, what sometimes you'll find is either a single or a small subset of compounds that are overexpressed and correlated with fertility. And so that's when I can go in and actually do bioassays and manipulate those single compounds within the overall profile and really test if the ants are perceiving that as a fertility signal or not. So sometimes we luck out, and sometimes just one, which is the case with the current species that I studied, the trap giant, Dynamacus bruneus. It's just one compound that is a fertility signal. So that's really just a luck thing that happened in this system. But other systems, it's more complicated because it is multiple compounds.
So when you figured out something like having one of these, if it's a single hydrocarbon and you're wanting to do a behavioral experiment, how do you actually go about doing that?
How do I go about doing the experiment or figuring out what compound it is?
Doing the experiment.
Yeah.
So are you wafting around a little scent and just seeing what all the ants do, or?
Well, over the last 3 years, I've formed this collaboration with a chemist at UC Riverside, Jocelyn Miller, and so he can actually synthesize these chemicals. And so they're really long waxes. They're not volatile. They're contact pheromones. So they're basically only perceived by the insects when they're in really close proximity to the other nestmates, or even touching the other nestmates. So what I'll do is I'll anesthetize an ant, and then I'll rig up some system where I can add this compound and supplement it to their naturally occurring compound, and then put the ant back in the colony and actually figure out and sort of gauge what her nestmates' behavioral responses are to her. And that's sort of the major sort of backbone of most of these bioassays, is trying to go in and manipulate the naturally occurring chemistry and the naturally occurring profile, and then really gauge what the nestmates are doing in response to those manipulations.
Do you tend to stick within single species or single populations, or do you look at what happens when you sort of go across different populations and species?
Yeah, so most of my work for my postdoc has been in one species, and that's mostly for the reason of, for fertility signals in general, we only know, we've only experimentally identified 7 fertility signals for ants out of all ants known. So it's sort of a burgeoning area of research, and the first one was done in like 2009. So there's a lot of work to be done within species. So I've had several studies within one species, and I've looked at a variation within population or between populations of this one trap giant across Florida where it occurs. But I've also the next paper I'll be working up is a comparative sort of study of all the rest of the trap giants in the southeastern United States. There's 3 more species in the southeastern U.S. And so it's a comparative paper between that. So I am branching out into sort of more comparative within the genus studies, but there needs to be a lot of work within single species too. Definitely.
Cool.
So I guess that kind of brings us to what your latest paper was on, where you were looking at the worker responses to queen pheromones, correct?
Right, exactly.
And then you were finding their behavior was kind of weird, and doing some unusual, human-centric, or what we would typically do to a queen, perhaps, is kind of bowing down to them in a way, right?
Yeah. So this species, they belong to a more, I'll use quotes here, primitive clade of ants. So they have more sort of ancestral traits, sort of wasp or bee-like social traits, where dominance and small colony sizes are really common within this clade of ants, and it's true for almost all trap giants. And so what these workers do when they're near the queen is they immediately adopt a submissive pose, which it's sort of exactly what you think of when an ant. If you think of what an ant might look like when it's submissive, that's what happens. They crouch down really low, and they sort of pull back their antennae and sort of cower away from the queen whenever they're close to her, the queen walks up to them. So that was, and that's actually one of the behaviors that I used to study. That's called a releaser effect to a pheromone. That just means a reaction, basically, to a pheromone, an instantaneous response to it. And so that's basically one of the main releaser effect response to these queen pheromones that I see in this species of ant.
And so, do you have any idea why they may be acting so submissive to it? Is there a risk that if they go in too boldly, that the queen will, act aggressively towards them to keep them in line, or is this just them, being, I don't know, manipulated by the Queen's cuticular hydrocarbons in some other way?
Yeah, so dominance is used a lot in this species, even between workers, not even between queens and workers. In 1999, there was an animal behavior paper done by Scott Powell that showed that dominance interactions in the forms of rapid antenation, where they'll basically box each other with their antennae, slap each other in the face repeatedly. The workers will do that, and that determines sort of the division of labor and the worker hierarchy within the colony. So the losers of those bouts get pushed out of the brood area of the nest, and basically out of the nest into the most risky tasks of a colony, which is going outside and actually foraging for food. So basically the least dominant individuals get pushed out for that. So in evolutionary terms, it makes sense that the queen, once she's actually reproductively dominant, once she's actually being reproductive, that she shouldn't need to sort of show her dominance through costly behaviors such as rapid antenation or actual physical dueling between workers. So that's when pheromones come into play. So the queen can actually signal her dominance, that she's already she's the reproductive. She's the one who's basically already built up the physiological means of actually laying the eggs in the colony. So she signals that through fertility signal to her workers, and the workers respond to that with submission instead of contesting another random nestmate in the nest.
And so when you released the paper, excuse me, so it just came out this week, and when you put out the paper, you actually took it a step further and actually produced like a video abstract / explanation to go along with it, which is something that is maybe not as popular to do by academics. Can you tell us a little bit about why you did it, what you wanted to accomplish by sharing your work in this different way?
Yeah, well, I don't know. I mean, it's kind of, I feel like it's kind of boring to just publish a scientific paper, right? I mean, I feel like unless it's a Science, Nature, PNAS paper, if it's like a a mid-tiered with quotes journal, like even a journal like Biology Letters isn't. It's a good journal. It publishes general papers, but it's not a journal that has every paper that comes out of it is guaranteed to get media attention, or there's I'm not exactly curing cancer or something like that, right? So I knew that this paper was destined to just go and be deposited in the annals of science, and the 10 people who worked in this field would see it and would read it, and that'd be it, right? And maybe it'd get 2 citations a year for X amount of years, and that just makes me sad inside a little bit, I mean, how much work goes into every paper, and then that's just it. We submit it to a journal, and then a couple people read it, but where's the work, right? So I knew that this paper was destined to obscurity in a sort of a general way. So I wanted to sort of, I'm interested in science communication, and I'm interested in how science communication plays a role in the perception of scientists in society, and sort of how that goes into their jobs, and how that, I feel like it matters for every part of our job. So I wanted to do an experiment with this paper where I basically, as the person who wrote the paper, did the experiments. I wanted to be the person that took the time to explain it in the most general sort of way, and in the most fun way I could do, and with the coolest videos that I had on hand for those ants. And I just wanted to present it so anybody could go and watch this 3.5-minute video and understand oh, there's the person who did the science. That's what he did. This is what he thinks what he thinks it means, and this is why he thinks just science is important in general. So instead of someone looking up the paper and just being confused because it uses a bunch of jargon, I wanted to have something that people could go to just to look it up and get an idea of what I did really quickly. So that was the reason. I mean, among many things, that was the main reason why I made the video.
I think it's an awesome reason, and I think the video was really, really well done. So we'll put a link up to it on the Breaking Bio website at breakingbio.com so that people can check it out if they haven't done so yet. But I mean, it was really, really well done, and I think it accomplished all those goals that you were just talking about there. Do you have any idea on, will you be tracking, let's say, like the response that it may have? Do you think it'll lead to more citations or, greater academic, qualifiers of research?
Yeah, it already has. I've already gotten paper requests from a paper, and I've never had paper requests like within the first week of a paper going online. Like, I didn't send it to anybody. But people have heard about it and seen the video. Mostly people who have seen it are other scientists so far, and I think most of them have been aware of the paper through the video, because that's basically my social network. Mostly is through Twitter, is mostly other biologists and other scientists. So I think even though I didn't really make the video for scientists, that's sort of been the audience so far, but that's been the audience because that is my social network, and that's the only way the video got distributed at all. So I think, I think if it's a different issue if the video reaches more of the public, because I feel like there's still some gatekeepers that need to actually present it to the public, and by that I mean, the more traditional sort of press release, university backing, send it out through the university's resources, or even through the journal's resources to the public. But that hasn't happened yet. But it might. But it was an experiment, like I said, just to see what would happen if I did this.
So where do you see something like this fitting into science communication? Is this something every scientist should be doing?
No, I don't think. I think science communication is important for scientists to do in general. I think everybody should do the form of science communication they feel best suited for. I don't think everybody wants to go out and make a YouTube video and put that on YouTube. I don't think every scientist should have a blog or a Twitter. I think that engagement with the public and communication of science and what scientists do should really be through what the scientist feels like they can express their voice mostly through. I'd be really bad at blogging because I'm a really bad writer. Like yesterday, I reopened my paper and I was like, did I spell one of the words in the title wrong? Because I really don't know how to spell. So I just had I had a really big freak out yesterday that I thought, oh, did I just publish a paper with a misspelling in the title? That's only 5 words long. I really thought about that. I had to go look it up, see if I spelled the word dependent wrong. I might have. That's just to say, like, there are certain forms of communication that I feel well suited for and comfortable doing, and they're definitely not all science communication. I wouldn't be good at most of it, but there are some things that I feel like I'm better suited for. Does that answer the question? I don't know. That's rambling.
Yeah, no, I think it does. I wonder if there's maybe a hybrid approach where, the press release offices of a university do the sorts of things that you just did with your paper, making lay summary videos and things like that, instead of just dumping a press release that says, oh, hey, he just cured cancer with ants.
Right. Yeah. Yeah. Well, I think that for me, that's one important thing about communication now, basically with the internet, is that the old model seems to be that press offices would really sort of judge your paper's impact and what people might want to get, what the public might want to get out of your paper, and then digest your paper and spit it out to news articles. But but really everybody has the means and sort of the mechanisms to put out their own message now. Like us as scientists, not as primarily communicators, but primarily as scientists, the communication part is more accessible now, I feel like, than ever, right? So that's more of an opportunity for us to actually have a voice and actually, because we have the insight itself, because we did the science, we know what it means to us, right? We know why it's important to us. So I feel like that puts us at an advantage in terms of what a press office can offer versus what we can offer if we can communicate sort of why we feel like what we do is important and why it's neat and unique and why people should value it. And so that's another sort of main motivation that I have when I do science communication is I feel like communicating the values that I feel are important in my work and that, you know, why other people might appreciate, you know, what I do in general or what we do at large.
So I think you articulated very well our reasons for doing this podcast, because we wanted to give scientists a platform with which to speak about their science themselves.
Yeah, exactly.
So maybe it's a good time for you to tell us a bit about your podcast and what it's about and what you're aiming to achieve with it.
Sure, yeah. Well, it's along those same lines, so it's the same motivational factors that I really wanted something to be out there that was an in-depth sort of look at these well-established scientists and get basically in their own words. Most of my interviews focus on a small subset of things, like how did the people get interested in science, how did they get to where they are today, what were the important things about that, and then where do they feel the true value of their job as a research biologist and as a scientist is. And I also sort of play up that I'm an active scientist. Like I'm a junior colleague to all these people that I interview. Colleague might be a stretch, but I'm junior to all these people that I interview. Right, and I want them to sort of basically answer those questions to me personally and to sort of younger scientists about, you know, what is the, like when you boil it down to everything, what is the core fundamental value of why you think what you do is important and why what we do in general is important. So I'm really just trying to get people to answer that question. And sometimes it takes an hour and I don't get an answer to that, but I try, and that's really the point of it, I think.
Yeah, I think it's really, really good. And again, anybody who hasn't heard of the Age of Discovery podcast, we'll put a link on our website so you can find it. Because it is, if you're a biologist, and like you said, if you're a junior biologist and a colleague or a future colleague of any of these people, I think you'll get a kick out of it. But you've talked to some fairly big, shiny names in the world of evolutionary biology in the last year and a half. So I'm just going through the list. You've got Walter Schinkel, May Berenbaum, Bert Holdobler, Jerry Coyne. I mean, these are big names, and you do a really good job of diving into their backgrounds and learning not only how they got interested into science, but also how they managed to get to this place that we've put them on, this pedestal of sorts that we have them on today, and about the work that they're currently doing. Have you had many struggles with getting in touch with these kind of people?
No, it's been, no one said no yet, but I have had to like pester people. So there has been multiple emails, and sort of the emails seem a little bit desperate, probably.
We know that feeling well around here, don't worry.
If you're on the receiving end of those. But I tried to like get across that like I've done a lot of research into the people that I'm interviewing. So I try to read as many of their books as possible, and I also try to target people who at least some aspect of their work that has had some sort of public communication aspect to it. So either they maintain a blog or have written a book, a popular book usually, or done something in the popular media, so that I sort of take a wild guess that if a person has done one of those things, like they are somehow feel that communication in general and sort of science communication and public communication is important. And so I try to get across that, like, I'm gonna ask you questions that no one has asked you. I'm really serious about it. Like, I don't do an interview unless I'm sitting in front of the person. So I say, like, I have to be in your office. Like, we're gonna talk, and I'm gonna drive up to see you, and we're gonna do this. And so I don't know. I try to get it across that, like, I'm putting a lot of effort into it, and I'm not gonna make them look bad, and it's gonna be something that they haven't done before, potentially. So that's been the angle so far.
You know, I noticed that a lot of the people that you're interviewing are senior, essentially, you know, in terms of position within the field. But I wonder, like, if you were, I feel like science is changing in some regards, in terms of career progressions, in terms of, you know, how things are going to look in even 20 years. You know, if you come back and you were going to interview Morgan when, you know, in 30 years when he's at the top of his field, you know, or has washed out completely, what do you think that would look the same as a podcast you're doing right now with these people?
No, that's the fun of it. It's I'm learning a lot because science has changed so much, and sort of the North American approach to university science is, like, changed so dramatically based on the time most of the people I interview got hired to today. So I've interviewed several retired professors so far, and it's mind-blowing, sort of, when they tell me how they got started and sort of their career trajectory based on what it is today. And I think that's part of it, and that's part that I want to. That's part of the reason why I go after these well-established people and these sort of senior and, in some cases, retired people, is I want to hear those stories, and I think it's a really important thing to consider for the future of science. Just sort of what the crop of current scientists we have, and sort of the leaders of the field and how they got there based on their sort of cultural context in which they got hired or started their careers, and how different that is from today. And sort of thinking about that for the trajectory of future research biology and the choices that a lot of people in our current generation have to make to sort of influence what's going to happen in the future for research biology and funding and hiring, all that sort of stuff. So yeah, there's several moments in my interviews where I'm just, like, dumbfounded in what they tell me about, you know, how they got hired or how they got tenure or what the job market was like compared to today. It's insane, just in one generation, how much things have changed.
Yeah, I know when I've been listening to it, I listen to them when I'm walking into work or walking home from work in the evenings, and there's been more than one instance of me shaking my head thinking, I wish that the situation was somewhat similar, because my measly 3 publications now would have got me tenure 50 years ago.
Yeah, right. Right.
It's such a different world. It's mind-blowing. But all of your guests have been really, really good about you know, opening up about their lives and their careers in ways that, like you said, you must have been asking the right questions that nobody else has, because they've all been really engaging interviews.
Thanks.
So who do you think, who would you like to get onto the podcast soon or coming up? I mean, you being an ant person and talking about public engagement, I got to assume that E.O. Wilson is fairly high on your wish list on this one, but maybe I'm totally wrong.
You are actually totally wrong. You know, E.O. Wilson, he does so many interviews. I feel like his life is out there completely. I don't know if I could ask him an original question, and
I can think of
something about group selection.
Yeah, but that's been all over the news. Like, he gets asked that all the time, right? And but also I should say that in my podcast I don't really ask people about research results. I don't really ask them about their findings or their discoveries, really. I'm more interested in their careers. So a lot a lot of my things go in. I talk about their careers and their lives rather than their research. But E.O. Wilson's a pro at interviews, so I'm not sure I could ask him a question that would be completely original, or that he would give me an answer that would be something that he hasn't said to another interviewee. But there are several people who I do have my sights set on. The next, I don't know if this will happen because he's a really busy guy, is Sean Carroll at Wisconsin, the Evo Devo guy. I've been going through his catalog, reading his books, and he's relatively close to where I am in Illinois. But I haven't reached out to him yet because I haven't done enough research for him yet. And then the next exciting thing is one of my favorite guests from the year and a half that I've done it was Eric Pianka. He's a lizard evolutionary ecologist, and he wrote the book Evolutionary Ecology. We hit it off, and he's got some crazy stories from his childhood and his career. And I'm going back to Austin in a couple weeks, and I'm probably going to do a follow-up episode with him, but I am for sure going to we're going to work on this project where he wants to record eco poetry, and then he wants we're going to collaborate on some YouTube videos of him reciting poetry, and it's going to be mind-blowing, and it's going to be awesome, and I'm super pumped about that. So there will be another episode probably with him, but there will 100% be some awesome poetry YouTube videos about extinct wolverines with Eric Pianka. So,
I'm sensing we get Tommy back on. We have ourselves a little poetry rap slam here.
Yeah, definitely, right? The world needs that. The world needs more poetry in general, always. So there you go.
All right, that sounds weird and amazing.
Yeah, exactly.
So maybe we can just finish off by putting the focus back on you rather than on the people you interview. So what's next in your research? What are you looking to work on in the future?
So I'm still working on trapjaw ants. Hopefully, as long as I'm in research, I'll be working on trapjaw ants for a while. Working on more comparative chemical ecology stuff. And I got to tell you, the science communication stuff, I'm one of the benefits of a postdoc is it's sort of where the rubber meets the road, and it's where you have to. I feel like I had to personally make decisions about what I wanted to sort of focus on in my research, and what I do now, and how that affects my long-term trajectory as a biologist and as a scientist. And I think the more I do it, the more important I feel that science communication is more making. For me, it's making videos, it's doing the podcasts, doing multimedia things. So more of that definitely in the future for me. More video projects, more weird poetry YouTube videos, all that sort of stuff, which I feel like is important and doesn't get emphasized enough in the general career of a biologist. But hopefully that will change.
Awesome. Well, I'm sure that there'll be many people at home listening right now that are looking forward to Wolverine poetry and everything else that you come up with, both academically and in the more public realm there. So where can they follow along with all of your adventures and possible future ventures?
Yeah, so you can go to my website. It's just my name, Adrian Allen Smith, ALAN Smith.com, and that has links to my YouTube page, my Twitter account, those boring old papers if you want to read those, and the podcast also. The podcast is Age of Discovery. It's AODPOD.com. Just anywhere on the internet you'll find me.
Awesome. And like I've said multiple times already this episode, we'll make sure that we have links to all of Adrian's stuff on our website, which is at breakingbio.com. So we're going to wrap it up here, I guess, and end it off for another week. But we need to thank the European Society for Evolutionary Biology for giving us some money to help keep us moving along. And if you want to drop us a line at home, drop us a comment, tell us what we're doing right or wrong, or suggest some more ideas on who you think we should talk to, you can do so on Twitter by messaging us at Breaking Bio. You can find us on Facebook, searching for the Breaking Bio podcast, and of course you can leave us a message or a rating at iTunes by searching for the Breaking Bio podcast as well. That's it for this week. I want to thank Adrian once again, and we'll talk to everybody again next week.